Hello everyone, welcome back to Lifehue YouTube channel. The topic for the day is histamine from pharmacology. In this lecture video, we are going to discuss about the histamine, its synthesis and destruction, storage and release, and histamine receptors. In part 2, we will discuss pharmacological action and pathophysiology of histamine. Histamine means tissue amine. The term histose means tissue. Histamine is present in almost all animal tissue. It is primarily within storage granules of mast cells. Non-mast cell histamine also occurs, such as in brain. The turnover for mast cell histamine is slow and non-mast cell histamine is fast. Histamine is present in most body secretions and pathological fluids like venoms, etc. Synthesis and destruction of histamine. Histamine is produced from histidine by an enzyme known as decarboxylase. It is degraded by either oxidation or amination. As you can see in this diagram, N-methyl transferase convert histamine into N-methylhistamine and diamine oxidase convert it into imidazole acetic acid. Storage and release of histamine is in mast cell granules mostly. As you know, histamine is a positively charged molecule. It is stored in mast cell granules where negatively charged proteins and heparin are present. When it comes to the release of histamine, granules are released in extracellular fluid where there is exchange of Na ions with histamines and histamine is released in its free form. Histamine receptors. There are four kinds of receptors of histamine: H1, H2, H3 and H4. They are G protein coupled receptors. The location of H1 and H2 receptors are such that H1 receptors are present in intestinal, bronchial, uterine, smooth cell, CNS, endothelial cell, sensory nerve endings, and H2 receptors are present on parietal cells of GIT, blood vessels, heart, and brain. The specific antagonist for H1 is 2-methylhistamine, 2-pyridyl ethylamine, 2-thiazole ethylamine, and a specific agonist for it is mipramine, chlorpheniramine. Mechanism of action for H1 receptor is that PIP2 hydrolysis leads to IP3-DAG and release of Ca2 plus ions from intracellular stores. Protein kinase C activation takes place. When there is increased amount of cGMP in cells, there is no release of H1. For histamine. For H2 receptors, diimaprid, imipramine are specific antagonists, and cimetidine and ranitidine are specific agonists. Mechanism of action is that adenylyl cyclase activation takes place. After that, CMP increases and phosphorylation of specific proteins takes place. This is all about the part 1 of histamine, where we discussed about histamine, its synthesis, release, storage, and also histamine receptors. In the next part, we will discover pharmacological actions and pathophysiological roles of histamine. Consider like, share and subscribe to my channel for more such videos.